Lysosome. As a result of microscopic studies, rounded dense bodies observed in liver cell. These bodies are initially described as pericanalicular dense bodies. In 1955, C.D. Dewey renamed these organelles as lysosome because they contain enzymes capable of lysis. A lysosome, therefore, is a lytic body. Structure and occurrence. The lysosome appear as a dense body in the cell and are surrounded by the membrane. The interior contains granulated stroma and vacuole. Isolation and chemical composition. Lysosomal fraction is isolated by centrifugation after mild homogenization. The fraction usually has contamination of mitochondria and microsome, but pure fraction of liver lysosome have been obtained in good quality. Each lysosome contains about 40 types of hydrolytic enzymes, proteases, nucleases, glycosidases, lipases, phospholipases, phosphatases, sulfatases. For the activity of these acid hydrolases, the lysosome maintain a pH of about 6 in its interior. If these enzymes leaks into cytosol, they can do little damage at higher pH there. The lysosome membrane also contains transmembrane protein which help in transport of the final products of digestion to the cytosol where they can be either excreted or reutilized by the cell. The lysosomal membrane also have hydrogen pumps which utilize the energy released due to ATP hydrolysis to pump hydrogen into the lysosome, thus maintaining the lumen at its acidic pH of approximate 5. Most of these lysosomal membrane proteins are highly glycosylated, which perhaps protect them from lysosomal proteases. Lysosomes. Types of lysosome. Primary lysosome. Primary lysosome is also known as starch granule. is a छोटी स्मॉल बॉडी होती है, राइबोसोम से सिंथेसाइज होती है और एंडोप्लाज्मिक रेटिकुलम से अटैच रहती है. जो ये फर्स्ट एंटर होती है गोल्जी कॉम्प्लेक्स में, वेयर एन एसिड फॉस्फेटेज रिएक्शन टेक्स प्लेस before they are transported to primary lysosome, which may be preferentially charged with one type of enzyme or other. These acid hydrolases are present only in secondary lysosomes. Using autoradiography technique, the transfer of protein was observed have the following sequence: ER, Golgi complex, lysosome. Secondary lysosome. Secondary lysosome, also known as heterophagosome or digestive vacuole, results from phagocytosis or pinocytosis of foreign material by the cell. It shows a positive phosphatase reaction which may be due to a association with primary lysosome. The engulfed material is digested by hydrolytic enzyme. The products of digestion pass through the membrane of lysosome and are incorporated into the cell. Residual body are formed if the digestion is incomplete. Residual body is removed by defecation in some protozoa. An autophagic vacuole, also known as cytolysosome or autophagosome, is a lysosome contain a part of cell itself for digestion. Plant and fungal vacuole occupying 30-90% of cell's volume are also comparable to lysosome of animal cells. They contain hydrolytic enzyme but perform diverse function including the storage of nutrients and waste products, protein anthocyanin, rubber, degradation of waste material, increase of size, control of turgor. Functions: endocytosis and digestion of macromolecules. The best studied function of lysosome is the digestion of endocytosed macromolecules. These molecules are initially delivered into early endosome having a pH of 6 and is completed in lysosome with a pH of 5. While endocytosis is a phenomenon common to all cells, phagocytosis is restricted to specialized cells called phagocyte, exemplified macrophages and neutrophils. Digestion of external particles. Large particles of microorganisms are taken into the cell by the process of phagocytosis. The cell engulfs the particles and then forms an invagination that becomes pinched off from the cell membrane and forms an internal sac. Such a sac is known as phagosome. The phagosome moves towards a lysosome and fuses with it from one structure. The enzyme present in lysosome will call digestive vacuole or secondary lysosome. The residues of digestion are left in digestive vacuole. The vacuole now move towards the cell membrane and a process reverse to phagocytosis occurs. Therefore, if the process of digestion has to continue, lysosome should be produced at the rate of phagocytosis at the rate of phagocytosis. This process of degradation of large particles generally occurs in cells specialized for phagocytosis of large particles and microorganisms. Macrophages and neutrophils and vertebrae are examples of phagocytes. इंट्रासेलुलर डाइजेशन का भी फंक्शन होता है लाइसोसोम का. लाइसोसोम क्या होता है? डाइजेस्टेड पेनिट्रेट द सेल्स ऑन लाइसोसोम. द प्रोसेस इज नोन एज ऑटोफेगी. For instance, the degeneration of tadpole tail is produced by the action of certain proteolytic enzymes called cathepsin contained in lysosome. Cellular digestion: when a cell dies, the lysosome membrane ruptures and the enzymes are liberated. These enzymes then digest the dead cell. This is an inbuilt mechanism of autodigestion to get rid of the cell debris. Extracellular digestion: sometimes a process reverse to phagocytosis occurs and enzymes form a lysosome and liberated outside the cell to which a lysosome and its enzyme belong. It is perhaps through such a mechanism that the sperm penetrates the protecting coating of the ovum. For digestion inside the cell, any material to be digested is surrounded by membrane to form a vacuole or what is described as an endosome. Certain food vacuoles has been described in plants which are analogous to lysosome in function. In order to absorb a large molecule, the cell wall forms an invagination which surrounds and encloses the molecules, thus forming an endocytic vesicle called endosome or phagosome. These organelles, as is stated, do not possess adequate enzymes to digest the enclosed molecules. According to one hypothesis, the vesicle travel and come in contact with Golgi complex and drives acid hydrolysis from there, so that Golgi apparatus should be primarily involved in the formation of lysosome. Lysosomes are exclusively formed from Golgi vesicle and the hydrolysis synthesized on ribosome would be subsequently transported through the involvement of endoplasmic reticulum.